Hi everyone, I'm Anitha, Assistant Professor from Department of Biochemistry. Today in this video, we are going to see about the topic Golgi complex. So what is Golgi complex? So Golgi complex is one of the cell organelle. It is a stack of membranous flattened sacs and vesicles concerned with cell secretion. It was first described by Camillo Golgi in the year 1898 in nerve cells. Other name of Golgi complex is Golgi body, dictyosome, lipochondrion, and canalicular system. Structure of Golgi complex. It is a stack of membranous flattened sacs and vesicles interconnected by tubules. The size of Golgi complex will be varies based on the species. It is larger and well developed in gland cells and nerve cells, and it is poorly developed in muscle cells. As the cells become older, it tends to decrease in the size. It is of 1 to 3 mm in length and 0.5 micron in thickness. The shape of Golgi complex varies from one cell to another. They may be in the form of rods, granules, vesicles, or network. Electron microscope study shows that Golgi complex is a disc-shaped structure formed by a stack of cisternae and a network of vacuoles, vesicles, and tubules. So in the structure, first part is cisternae, which is otherwise called as lamella. The cisternae are elongated, flattened sacs of Golgi complex filled with fluids and piled one above the other to form a stack, and they are arranged in a parallel bundles one above the another. In most of the cells, the number of cisternae will be varies from 2 to 8. The adjacent cisternae are cemented together by a cementing substance called intercisternae material. In certain cases, the cisternae contain pores and they are said to be fenestrated. The cisternae are slightly curved, hence the cisternae have convex and concave surfaces. The Golgi complex has two slides, namely forming phase and maturing phase. The convex surface is a forming phase or cis phase. This phase is towards the nucleus or endoplasmic reticulum. Here a new lamella will be added from the endoplasmic reticulum. The concave surface is a maturing phase or trans phase. The trans phase is towards the plasma membrane, have large secretory vesicles called as where it is butted off. Thus, the cisternae are continually receiving the lamella on the forming phase and losing membrane on the maturing phase through the formation of secretory vesicles. So second part is vacuoles. So they are large specious round sacs, and third one is vesicles. So these are small droplet like structure about 40 angstrom in diameter. And next part is the tubules. From the trans phase of cisternae, many small tubules are arises and they interconnect the cisternae, vacuoles and vesicles, and they transport substances between them. Next we move on to functions of Golgi complex. So in that first one is formation of acrosome. Acrosome of sperm is developed from Golgi apparatus during spermatogenesis. During spermatogenesis, a vacuole appears in the Golgi complex. So inside the vacuole, there will be a dense granule will be formed, which is called as proacrosomal granule. Once the proacrosomal granules are formed, simultaneously, vacuole, both proacrosomal granule and vacuole will be started enlarge. The entire apparatus then move towards the nucleus and it will be attached to the tip of the nucleus. Then the granule now is called as acrosomal granule, and the whole structure, that is an entire structure, is said to be acrosome. And second function is cell wall formation. Golgi complex is involved in cell wall formation in plant cells. So during cytokinesis, the Golgi vesicles accumulate in the equatorial plane and help in the formation of cell flake. The material present in the Golgi complex help in the formation of cell wall. The polysaccharide of cell wall is formed in the Golgi complex. And third function is plasma membrane. Golgi complex involved in the formation of plasma membrane during exocytosis. The secretory vesicles formed from Golgi complex fuse with the plasma membrane. The membrane of the vesicle became incorporated into the plasma membrane. Due to exocytosis, the surface of plasma membrane increases. And fourth function is biogenesis of lysosomes. So genesis means formation. So Golgi complex is involved in the formation of primary lysosomes. The cisternae of Golgi complex in turn bud off small vesicles called as primary lysosome. This primary lysosome again fuse with pinosomes or phagosome to form secondary lysosomes. And fifth function is membrane trafficking. Golgi complex carries out a trafficking function in the transport of biosynthetic products, and they are transported within the cells. It may be intracellular or intercellular transport. And last function is sulfation. It is Golgi complex mainly involved in the metabolism of sulfate. So this is about Golgi complex.